Okay. Hello, everyone, and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Itamar Ronen. Dr. Ronen obtained his PhD in physical chemistry from the School of Chemistry in Tel Aviv University, where he worked with Professor Gil Navon on developing a method for indirect NMR detection of oxygen-17. Following a postdoc fellowship at the Center for Magnetic Resonance Research at the University of Minnesota with Dr. Sonji Kim and Dr. Dashi Kim, he obtained his first academic position at the Boston University School of Medicine, and there, together with Dr. Dashi Kim, he co-founded the Center for Biomedical Imaging and a master's degree in bioimaging. In 2009, Itamar moved to the Netherlands, where he joined the C.J. Gorter Center for MRI at the Leiden University Medical Center as a PI and as an associate professor, focusing mostly on developing methods for diffusion of intracellular metabolites in humans at ultra-high field, and recently developed spectroscopic techniques suitable for low-field MR as low as 0.05 T. Since October 21, he holds the position of academic director of the Clinical Imaging Science Center and chair in medical physics at Brighton and Sussex Medical School in Brighton, UK. Very exciting. I would just like to remind the audience to please address any question you have using the Q&A box or just raise your hand. Dr. Ronen, thank you so much for coming here today. The virtual stage is all yours.
Thank you so very much all, and thanks so much to all the attendees that took time from their lunchtime and joined this presentation. And I hope you enjoy it. I looked already at the list of attendees. I'm always curious to see whom do I know, because I'm an ex-Bostonian myself and so many familiar names. So I'm very, very pleased to see those familiar names and also make acquaintance with new names and new faces. So great to be here. And thanks so much for inviting me all. Without further ado, the topic of today, I'll try to be focused on one topic. I asked Ori if he wanted a little bit, also from the low field stuff, but then eventually I thought it would be nice to be coherent and not to jump around too much. Mostly would focus on diffusion-weighted magnetic resonance spectroscopy. I called it the inside story, even though one can say that there are some exceptions to that statement, but you will probably see what I mean by this not particularly successful pun. Let me start the laser pointer so that it becomes useful later on. And just quickly about what I'm going to talk about today. So I will remind those, I saw that a video is in the audience, so I don't have to tell him about spectroscopy, but certainly there are people that are not familiar with magnetic resonance spectroscopy on a daily basis. So I will just remind people of the advantages of getting rid of the big signal of the water and focusing on those tiny little things beside this huge peak. And why do we want to use spectroscopy and combine it with diffusion and go beyond what the magnificent tool of diffusion-weighted MRI already gives us today. I will give a couple of examples, not only from my work, but also from other people's work about unique microstructural information that can be derived from diffusion-weighted MRS. I will talk about a topic that is particularly close to my heart. I'm all about multimodal merging of information, and I think that there is no silver bullet in any type of method, and it's important to look at things from different perspectives and to talk about the benefits of taking information that has a high specificity, like the one that we get from diffusion-weighted MRS, but also to go to high sensitivity methods like diffusion-weighted MRI and combine them to get a larger picture of tissue microstructure. I'll give a couple of clinical examples. I'll first try to convince you that there is a point in looking at clinical or clinically relevant phenomena with diffusion-weighted MRS. And I will focus on two aspects of clinical aspects or pathological mechanisms. One that has to do with neurons, one that has to do with glia, and of course, both of them have to do with both. But MRS allows us to accentuate this statement by looking more at neurons and glia as we want. And I will even try to convince you that there is also something interesting about diffusion-weighted MRS when it's implied or applied to look at brain function. I'll show you one experiment. I'll skip the other experiment that I thought I would have time to, but from experience, I don't. But I'll be glad to talk about it more extensively if there are questions about it. I do want to mention a workshop that we had here in Leiden on diffusion-weighted MRS, the so-called Lorentz workshop, a mechanism that I recommend everyone to try to use. It's an entity within Leiden University that allows to create cost-free workshops. Essentially, the organization pays for the facility and the workshop itself. And we dedicated the workshop to best practices of diffusion-weighted MRS. And I want to mention a couple of the outcomes of this workshop later on. But first to business after, of course, and I'll finish with some random thoughts that may have been collected throughout the presentation. But first and foremost, of course, to the science at hand. And at first, I want to convince you that there is a reason why we impose on ourselves this extremely difficult experiments of diffusion, combining diffusion weighting with magnetic resonance spectroscopy. And as you may know, as I think all of you know, that magnetic resonance spectroscopy is one of the most unique types of measurements that we can get, because it gives a window to brain neurochemistry without opening the brain and doing histopathology or immunohistochemistry. So we can look at the brain using magnetic resonance spectroscopy, which means that we get rid of the water signal and we focus on signals that are typically at the kind of right-hand side of the water peak, as we typically look at the spectra this way. And those peaks that are typically in amplitude, something like 3 orders of magnitude smaller than the water, just because of the concentration is much lower, represent hydrogen atoms that are part of molecules that are typically in the millimolar concentration. And those peaks are readily identifiable on the spectrum, and those that do not identify don't have to work too much, because we typically already know what is the content of neural tissue, and we know how to assign these peaks to spectra of individual molecules. And those molecules, in, let's say, in mid to ultra high field, we can get anywhere between 5 to 15 such molecules well represented and well quantified, are not evenly distributed across cell types. And of course, there is still a lot of discussion and debate as to what is exactly the distribution of those molecules across cell types. But some things I think we can say with a reasonable amount of certainty. And that's, for example, that there are molecules that are more found in higher concentration in neurons. Most famously is the largest peak in the proton spectrum of the central nervous system, and this is the combined peak of the N-acetylaspartate and N-acetylaspartyl glutamate, mostly NAA, and this goes under what we call a neuronal marker. It has, of course, also reasons why it is in neurons, but we typically refer to it as a neuronal marker. There are also the most commonly found excitatory neurotransmitter, glutamate, and inhibitory neurotransmitter, GABA, and other peaks that are considered to be predominantly neuronal. There are other metabolites that reside more in glial cells. What glial cells? One has to be a little bit more specific. We have at least 3 major types of glial cells in the brain. We have oligodendrocytes, and we have astrocytes, we have microglia. It's important to make this distinction. We don't always do, but we know, for example, that myoinositol, which is a kind of a sugar, is an osmolyte that is found mostly in astrocytes. Choline compounds, at least 3 of them, are also tending to be found more in glial cells, and here I'm bit more vague in my definition, but are also found in neurons. It's only that their concentration is probably higher in glia. We will see evidence for that later using diffusion-weighted MRS, which is kind of nice to see it coming from a surprising direction. And there are also metabolites that will come very useful later on. And this is the pair creatine-phosphocreatine, which is very important in terms of oxidative metabolism in cells. It is reasonably equally distributed across cells. But we will see that it also plays a role in measurements that we do using diffusion-weighted MRS without being particularly cell-specific. What defines all of those metabolites that I've mentioned so far is that in the healthy brain, they're all being kept rather strictly in the intracellular space. There are also metabolites that are found in the extracellular space, like glucose, and in the disease brain, you can find lactate also in the extracellular space. But those particular metabolites, either via active pumping or by the fact that they do not cross the cell membrane, are kept intracellularly. And the use that we do with these metabolites is essentially to paint our diffusion-weighted world in such a way that if we look at the at tissue as seen through the water protons, it's a rather monochrome picture whereby water exists in all cells and in all tissue compartments. And in addition, it also exchanges across tissue compartments. So it's a very sensitive picture, but it's not particularly specific. But if we are able to, for example, eliminate the extracellular space and also color code our cells based on their metabolitic content, then we add a layer of specificity while losing some layer of sensitivity. And with this specificity in mind, we can ask ourselves different types of questions that can be answers with simple tools. So the very same way that we add diffusion weighting gradients or imaging sequences can be done also to our spectroscopic sequences. This is just, these are just two very simple examples. This is the STEAM sequence added with two diffusion weighted gradients, the dephasing and the rephasing gradient in the half echo times on the two sides of the mixing time in this sequence. And here is a bipolar diffusion weighted sequence based on the press sequence. So these are just simple examples. And if we have those tools, then we can ask in a more compartmental specific way all the typical questions that we ask ourselves when we do our diffusion weighted experiments, only this time geared to get answers that come from, let's say, a more known address in tissue. So for example, we can do what we also do in imaging, time-dependent diffusion measurements to look at compartmentation. And for example, if we are looking at an intracellular metabolite that resides in a completely confined space, we would expect to see the time dependence of the diffusion coefficient to go down to 0 at very long diffusion times. And if we are in a tortuous space, like for example, in this case, this artificial or interstitial space, we would expect to see at very short diffusion times something that is close to the free diffusion coefficient, but then to reach the tortuosity limit and be banging against the cell membranes in the outer side of them. That meaning that at long diffusion time, we can diffuse to very long distances, but still with a kind of a hindered limitation by those structures. We can also look at anisotropic structures like fibers. And in this case, of course, our diffusion measurements will be directionally dependent in the same way that it does in imaging. But in this case, we know exactly the source of the time dependence. So for example, if we measure perpendicular to the fibers, and we have highly organized fibers that go in one direction, and we manage to find a very specific direction that is perpendicular to them, we get a very similar time dependence to the fully constricted space. And if we are measuring parallel to it, and I added those little red dots to create an intrafibronal tortuosity, then we have a very similar behavior to the extracellular space here. And then we can ask all sorts of interesting questions that we can then answer in a more cell-specific way or compartment-specific way. We can talk about cytoplasmic viscosity and tortuosity. We can look at cellular morphology, all dependent, of course, on the time that we allow to diffusion to act. That means that we have to be mindful also of the diffusion coefficients that we expect to see in the intracellular space, something that I will get to in a second. We can hope for looking at subcompartmentalization and even at compartmental exchange. And if we are really, really optimistic, we may even think about looking at chemical exchange and metabolism within those particular metabolites that we're looking at, and other things that can come to mind. Two things to bear in mind. One is the concentration of metabolites, again, much, much smaller than that of water. And that means that something has to give. And what typically gives is the spatial resolution. So we're typically doing experiments on volumes that are several cc's. And that means that everything that has to do with macroscopic anisotropy is typically contaminated by partial volume, by macroscopic curvature within the volume. So we have to think about what we really want to measure within our measurements. And that will come, I hope, a little bit clearer as the presentation goes further on. And another thing to be considered is that the diffusion coefficient of metabolites, even if measured in phantoms, is 3 to 4 to 5 times lower than that of water. And that's a blessing and a curse. It is a curse because if we want to get the effect of diffusion weighting in such a manner that you can calculate your error propagation and calculating, for example, diffusion coefficients is not too high, then you want to create an enough decrease in your signal when you do your diffusion weighted measurements, meaning that you have to go to B values that are about 3 or 4 or 5 times as high as the one that we use for water imaging. And that is, from the gradient perspective, can be a limitation. So if you don't have enough oomph in the gradients, we have to increase our echo time. You increase our echo time, you lose your signal, and so on and so forth. So that's the curse part. The blessing part is that the time dependence becomes more stretched. And if the time dependence becomes more stretched, then the amount of microstructural information that you get from the time-dependent portion of the diffusion coefficient of metabolites is bigger than what you can find with water, that becomes pretty much time-independent at very, very short diffusion time. So there is a situation where we can gain some, we can lose some, and we have to plan our experiment bearing all of these things in mind. And I'll give you a couple of examples, starting with the historical example, actually one of the first examples in which diffusion of metabolites was used to kind of provide some sort of a valid microstructural information. And this is the experiment in which Robin de Graaf, the Dutchman who is now sitting in Yale for many, many years, but at that time working with the late Klaas Nicolai in the medical center here in Utrecht, measured the time dependence of phosphorus NMR in phosphorus-containing metabolites, not in the brain, but in muscle. And the experiment is simple. Take some muscle from an animal, in this case, it was rat skeletal muscle, and perform measurements in parallel and in perpendicular to the muscle fibers. And these are just figures of the decay of the signal. And the biggest peak is the one of phosphocreatine. There are also peaks related to other phosphorus metabolites like ADP and ATP and so on. But the biggest peak is the one that is easier to measure. You see how in the measurement parallel, the signal decays much faster than in the measurement perpendicular. And when you take the average of 3 such measurements perpendicular to one another, so to calculate the rotationally invariant apparent diffusion coefficient, at very short diffusion time, you get a measurement that is very close, let's say, to the tortuosity limit, and maybe even close to the free diffusion of phosphocreatine at the temperature in which the measurement was done. But as the diffusion time increases, so does the the diffusion coefficient decreasing. And eventually we reach a plateau, and this plateau is reached when the diffusion coefficient is about the third of the value that you see at very short diffusion times. And it's not a coincidence. There is a rhyme and reason for that. And this is because if you can imagine yourself measuring in three perpendicular directions, a set of the diffusion in a set of cylinders, let's say that they are perfectly aligned with one another. If you're perfectly aligned with your gradient direction to the direction of the cylinders, then you essentially measure the free diffusion coefficient of what diffuses within those cylinders. And at the two perpendicular direction to it, you measure essentially a fully restricted diffusion, in which, as we saw in the previous slide, the diffusion coefficient is time dependent and will eventually reach 0 at very long diffusion times. But since we have three directions, two perpendicular and one parallel, then the weighted average is such that the average here is roughly the third of the value here. And from here, one can actually predict also the geometry of those fibers, which in this case, interestingly, it did not coincide with the main fibers that you see here in terms of their diameter, but there is an interesting explanation that is in the article, and I recommend you guys to read this very beautiful and historic article. Many years later, Julien Valette in Paris decided to do the same measurement in brain metabolites and look at what happens, the time dependence of diffusion coefficients of brain metabolites as one increases the diffusion coefficient and the diffusion time. And this was done using a steam sequence. And the question was a very simple one. Do we have in the brain any compartment that is fully restricted, like red blood cell, for example, something that is that can be assumed to be restricting diffusion at any direction in kind of an isotropic manner, as compared or as opposed to fibers, or what we call neurite, in which we can assume the system to be of cylindrical symmetry. So essentially answering the question whether the diffusion coefficient will reach a certain plateau for those metabolites, or will reach 0 in the case that we have some sort of an equivalent of a spherical cell body or cell type. And the answer was surprising or not surprising, depending on who you ask. If you ask a neuroanatomist, who would say that's kind of a trivial answer. If you ask an MRI physicist that always assume everything to be spherical, then there is a surprise element in it. And the answer was that the first experiment done in monkeys showed no decay of the ADC towards 0. So no such fully restricted compartments in the monkey brain. Then Julien decided to contact us, and together with Francesca Branzoli, who was then a postdoc here, and Chloé Najak, who was then a PhD student of Julien, we did the same experiments here on humans, and also did not see any decay of the signal to 0 ADC. So again, no evidence for a fully restricted compartment, at least between glials and neurons in the cerebrum in humans. And he repeated the same experiments also in rodents. And this is a very nice representation of results from 3 species, so monkeys, humans, and rodents, and at a very broad range of diffusion times, including extremely short diffusion times obtained by combining oscillating gradients with the spectroscopic sequence. That's not something that we did here, but he did on his animal scanners in Paris, showing that there is time dependence, but the time dependence is limited to extremely short diffusion times, as expected from an organ in which the predominant feature or microstructural feature is neurite, or meaning fibers. So very strong time dependence, very short diffusion times, but very weak time dependence at long diffusion times, showing essentially no fully restricted compartments. The other experiment that I want to show you is actually one that I started when I was in Boston University, and one that kind of brought me here to Leiden. And it was kind of more of a proof of concept, but it was a proof of concept that gave us an interesting insight that would also interest people that are just using spectroscopy without any relation to diffusion. And the idea was, okay, we have a human scanner, why don't we go to an area where we have a very strong macroscopic anisotropy, in this case, the corpus callosum, and repeat the type of experiment that Robin de Graaf did in muscle fibers, but to do it on a bunch of organized fibers in the human brain. And in this case, we started at 3T, but all the results that I show you here is from the 7 Tesla here in Leiden. Let's apply a strong gradient in the direction that is left-right, so parallel to the direction of the propagation of the callosal fiber in the human corpus callosum, and one that goes perpendicular to it. And here are just typical spectra that we obtain from one subject, two B values, one measuring in parallel to the corpus callosum, one measuring perpendicular to it. And when you organize the results for the 3 major metabolites, 3 major singlets on which we get very reliable data in diffusion-weighted MRS, these are the results for total creatine, these are for total choline, and this is for total NAA. You see that the measurement in parallel to the callosal fibers, the decay of the signal is very strong, and perpendicular, it's much, much more moderate. And then one can calculate those diffusion coefficients in perpendicular and or in parallel, and calculate something that I would call a poor man's fractional anisotropy within this huge spectroscopic volume. And you can see something that really gleans out really, really strongly from this, these results is when you take the ratio of the diffusion parallel and perpendicular to the callosal fibers, the NAA has a much more or much stronger directionality of diffusion than the creatine and the choline. And not only that, but also you can see that there is a kind of a gradation here. The most directional is NAA. Why? Well, contained only in axonal fibers that are the reason why we have macroscopic anisotropy in white matter. But creatine and choline also exist in glial cells that in themselves, of course, have fibers, but they do not necessarily follow the direction of the callosal fibers because they are essentially isotropically arranged in such a way that they have no macroscopic anisotropy per se. So the glial contribution is the one that decreases the overall macroscopic anisotropy that we see in the diffusivity of the N-acetyl aspartate. And you can see this kind of strong differential effect of macroscopic anisotropy, but that kind of gave me a kind of a simple idea to say, well, if we assume that the NAA is 100% axonal in the corpus callosum, then any decrease in macroscopic fractional anisotropy would be the contribution of glial cells. And that means that we can, if we assume that the NAA is 100% axonal and axons are the sole contributions to the fractional, the macroscopic fractional anisotropy, and the FA that comes from the glia is 0, then the axonal fraction of metabolite will simply be the ratio of those numbers to the number given from the NAA. And those numbers, I kind of, again, just for the sake of conversation, calculated here. And you can see that creatine seems to be kind of 50-50 between the axonal volume and the glial volume without saying anything about, what do we have more, axons or glia in the corpus callosum. So it's not a quantitative evaluation here, but at least it gives you an idea of the fact that, for example, we have a very strong contribution of choline that comes from glial cells, more so, for example, than creatine, but that also creatine is indeed present in both cell types. So it's kind of an interesting indication to the amount of glial concentration of metabolites without doing anything invasive or cell culture here. So it's a kind of an interesting result that has nothing to do with diffusion. Going back to the presence of metabolites in neurites, and the fact that we found that those metabolites are indeed present for the most part in neurites, and knowing that NAA is essentially a completely neuronal metabolite, Henrik Lundell from Denmark came up with the idea of, well, if this is the situation, then why don't us apply a very simple model that can take us away from the macroscopic anisotropy and bring us to what really is useful in large volumes, such that we measure in diffusion-weighted MRS, and this is the so-called microscopic anisotropy. And if there is a model that can bring us data and take us from macroscopic measurements and project into microscopic properties, that's of course a very important things to apply in this type of measurements. And the idea, of course, is an age-old idea that came from Callahan from the 1990s, and then further developed by Dmitry Yablonsky, for example, for measuring the diffusion of inert gases in the lung. And the idea is that if we have a situation whereby we have a fully dispersed neurite or a set of cylinders that are fully dispersed, then there is an analytical model to give us the dependence of the signal as a function of b-value on microscopic features of the anisotropy of those fibers. So the longitudinal and transverse diffusivity here refers to the diffusivity along each individual fiber. And this is, of course, a way for us to get rid completely of the dependence on the macroscopic world and focus on the microscopic features. Well, we're not always able to do this because we have a situation whereby, of course, in white matter, for example, we have a macroscopic anisotropy that is dictated by the fact that those axons run together in white matter tracts. But there is a trick. If you want to disperse those fibers, and you cannot do it on the system, you can do it in the measurement stage. And that is done by measuring the diffusion from multiple directions and averaging those results per b-values, thus losing the directional property of the measurement at a given b-values. And we did this experiment. We're using 12 directions with 5 b-values. If you want 5 shells of each containing 12 diffusion-weighted directions. This is, for example, 2 measurements that we did in the corpus callosum and in just random, not random, but a kind of a non-particularly distinct white matter portion in the parietal lobe. And I'm very, very proud of these spectra. Why am I proud of these spectra? Because you can see that these spectra retain their quality when you go to very high b-values, in this case to about 14,000 second per millimeter square. You can still see the shoulder of the NAAG on the pair of NAA-NAAG, which means that we are able to account for lots of spectral distortion factors like eddy currents and other factors by carefully doing these measurements and processing them properly. And once you do this and you fit your data to the very simple model that was suggested earlier, you first of all see that no matter where you are in white matter, you get eventually a curve that you can fit with this formula quite tightly. You can see that the spread, directional spread in the corpus callosum is much higher than in the parietal white matter because of the directionality of the fibers in the corpus callosum. And eventually, you get with a set of numbers that is quite consistent with each other and with additional measurements that I will give you quite soon after I finish with this discussion. And we, for example, find out that the longitudinal diffusivity of NAA is in the order of 0.5 micrometers square per millisecond, from which, by the way, one can calculate all sorts of interesting intraaxonal properties, for example, the intraaxonal tortuosity, to which I will get back a little bit later on. I want to switch gears and to come to a couple of measurements in which we combine data from water and from metabolites to give a bigger picture. The first measurement that I want to give you is something that I owe very much to Matt Buddy from Medical College, Wisconsin, who actually saw a poster that I was standing nearby in one of the ISMRMs many years ago. And he said, well, you know, that's interesting. Why don't we work together on trying to validate what you see here? And if you remember the measurements that I did on the corpus callosum earlier, I wanted to take them a little bit to the next level. And what is the next level? The next level is, well, you know, those measurements. It's nice to see that we get big differences if we measure parallel or perpendicular to the callosal fibers, but microscopic information we don't have yet because there is no model to fit the data to. And one of the things that kind of hinted to me that we need a model is, of course, what would have happened if those fibers ran really parallel to each other. In this case, we would have expected that measurement perpendicular to the fibers would result in almost no diffusion effect, and parallel would give us something that is close to, well, let's say the cytosolic diffusion of NAA, something that is close to this 0.5 that I showed you in the previous slide from the work of Henrik Lundell. But when you calculate the slope of those data, it's very far from both no diffusion and from the cytosolic diffusion of NAA. So something has to improve, or a model has to account for it. And the model has to account for two things. One is the fact that this is a very big volume, and the microscopic curvature represented here by the direction of the ticks, which is essentially the direction of the main eigenvector in the DTI measurements that I kind of superimposed on the spectroscopic volume here, gives us a kind of a range of directionalities that strays away from measurements that are purely perpendicular or parallel to the gradient direction. In addition to that, there is the orientation dispersion at the microscopic level of the fibers, which blurs even more the directionality of the fibers in the corpus callosum. And that calls for a model, and eventually a model came, and the model came in my feverish brain, very simple model, essentially one that takes the distribution of directionalities from the DTI data within the spectroscopic volume to account for the macroscopic curvature within the volume, and leaving a simple distribution parameter for the orientation dispersion that will account for the microscopic misalignment of fibers within any white matter tract, but also in the corpus callosum. This model essentially ends up with two free variables. One is the cytosolic diffusivity of NAA, and one is the variance or the standard deviation related to the orientation dispersion, the microscopic orientation dispersion. Came up with two numbers. One is 0.51, which we already saw in Henrik Lundell's work a couple of slides back. So that was encouraging. The order, by the way, was different. I first did these experiments, then Henrik came on, but it was good to see that those two numbers coincided. And then came this number that I had absolutely no clue whether it is a true number or not. And Matt Buddy said, okay, I can measure that for you. And he obtained brain slices from the Brain Observatory in UCSD and did an optical estimation of the orientation tensor, if you want, within voxels that are roughly the size of the DTI voxels that I used in my diffusion tensor experiment. And he came up with a number that is completely based on histology, which coincided, I should say, serendipitously with a number that we obtained with a very simple model that fitted the data just out of the diffusion of the NAA. So that just tells you that you can use very simple models and sometimes they work, but the take-home lesson here is that the model has to be based on information that comes from DTI as well as from the diffusion-weighted MRS, gives us something that is quite realistically and verifiable with histology. It would be a sin not to mention the paper that came up a few years earlier by the group in Norway with Lergaard and his colleagues that measured very similar orientation dispersion in the callosal fibers in the mouse. And you see that, yes, it is a little bit smaller, but we are still in the range of quite surprisingly high misalignment of fibers even in a very highly organized white matter tract such as the corpus callosum. From here, I want to bring you to a little bit more complex experiments. I hope to be able to talk about this experiment in the time that remains for me to go. And don't hesitate to just wield the axe and tell me this is the time that you have.
Take your time, Itamar, everything's great.
Everything is okay.
Yeah.
You know, I can go on forever. That's my trademark. So please don't hesitate to eventually put an end to this misery of the audience, and just also that remains also sometimes for questions. Already in the 1990s, Partha Mitra told us that there is much utility in trying to think about diffusion-weighted experiments that combine sequentially measurements in two different diffusion-weighted conditions. And one of the special cases of this type of measurements is the so-called double diffusion encode, in which we have two diffusion-weighted modules, one in which the direction of the diffusion-weighted gradient is fixed, and the other one that comes right after it, either immediately after it or allowing for mixing time if we wanted to, in which the direction of the diffusion-weighted gradients changes or rotates around the circle that contains the original diffusion direction, but strays away, becomes perpendicular to it, and one can complete the entire circle going from 0 to 360 degrees in sequential experiments. And the trick for why is it so useful? Turns out that if we have a situation whereby we have a dispersed kind of neurite like what we saw before, or in, let's say, a mathematically defined way, a set of diffusion tensors that are randomly oriented in space, but they are tensors that are anisotropic tensors, then the end result of the diffusion measurements at the end of those two modules will depend on the angle between those two gradients in such a way that we get a very beautiful modulation of the signal as a function of the angle between those two gradients. This is because, well, there are many ways to explain it, but one of the reasons that is easier to explain is the fact that if we apply our diffusion measurements to such a system, we kill very easily the diffusion that is parallel to this direction. And then if we try to apply it again, we have almost nothing that will feel the gradient parallel to it, but the tensors perpendicular to it will refuse to decrease in amplitude, and this gives rise to the strong signal here. But if we measure in perpendicular one time parallel to a set of fibers and the other time parallel to another set of fibers, then we get the signal quite heavily diffusion weighted. And that gives rise to modulation that is angle dependent in this type of measurement when we have an anisotropic set of tensors. But if we have a distribution of isotropic diffusion tensors, we will not have any such modulation. And this gives, again, the possibility from the depth of this modulation to estimate the longitudinal diffusivity within the microscopic features of our sample or tissue in this case, and the perpendicular to it, and to calculate the so-called microscopic fraction on anisotropy that can be zero if those fibers, for example, are randomly oriented, but it is above zero if we have fibers in the sample or in tissue. One of the main proponents of this type of measurements is Noam Shemesh, sitting in the Champalimaud Institute in Lisbon. And there he performed beautiful experiments of double diffusion encoded MRS measurements, showing that there is a very strong microscopic anisotropy, not only to neurons, as you can see here, but also the myoinositol that has 0 or very low concentration in neurons, but very high concentration in astrocytes, gives also rise to a microscopic anisotropy that is almost as strong as the one that we measure in neurons. And this is something that tells you that even in astrocytes that we can imagine, you know, in our kind of simple-minded MRI physicist eye as a balloon with fibers. No, it's mostly fibers. And the cell soma contribution is relatively small volume-wise. And we were highly interested in applying this kind of methodology also in humans. In humans, one can ask many different questions. For example, what is the difference when you measure it in white matter versus gray matter? And the other question that one can ask is, well, if you do this type of measurements also in water, then how does the measurement in metabolites relate to the measurement in water, not just in water, but in water in ever increasing B values? Because works that are accumulated experience is such that with ever increasing B values, the extracellular space is attenuated relatively much more so than the intracellular space. So we get a kind of a specificity by increasing the B value in a water-based measurement, or we can look at the chemical shift specificity, if you want, of the diffusion-weighted MRS measurement. And not to dwell too much on those measurements, I'll just show you the results. So one result that you can see already here is, if you look just at the spectroscopic data, you have a very strong modulation in white matter of both the glial as well as the neuronal metabolites. So we have a very strong microscopic anisotropy in white matter, nothing surprising here, but also in gray matter, except for the choline. Now, why is it that the choline, and this is kind of a bar graph that tells you roughly the same information. Why is it that in the choline we get much stronger variation, but also an average lower microscopic diffusivity? Well, the critics, including me, would say, well, you also have an SNR problem. And you can see even here in the spectrum that the choline signal from gray matter is much lower compared, for example, to the creatine than what you have in white matter. But I have to tell you that I also have the optimist in me that says, well, it might also say something about astrocytes in gray matter versus white matter. And I'm saying this because we also have an additional set of measurements that I'm not mentioning in this talk that points to that direction, that tells us that maybe the complexity of structures that contain choline, i.e. protoplasmic astrocytes in gray matter, may be part of the reason why we see this type of diffusion microscopic anisotropy difference in gray matter versus white matter. Against me is the fact that the creatine doesn't show this type of effect, but I am keeping this as a big question mark to be investigated more thoroughly in the future. And this is just to show you what happens when we compare the data to the water, which I think is a very cool result. So in white matter, when you look at the microscopic anisotropy of water, as it is changing when you increase the B value from something that is around 800 all the way up to 7,400, you see that the change in microscopic anisotropy is not that big. And this is a kind of a hint to tell us that if there is a strong contribution of extracellular water to the measurement at low B values, then also on the microenvironment level, the extracellular space is also anisotropic. And it kind of makes sense because if you pack fibers, then the extracellular space in the packed fibers in the microscopic environment might be indeed also anisotropic. In gray matter, the picture is completely different. At very low B values, the microscopic anisotropy is very low. But as we increase the B value, it increases to levels that reach the microscopic anisotropy of white matter. Not only that, but also the microscopic anisotropy of metabolites. So everything that we see here clumped in the very high B value of water and the B value in which we perform the measurements of metabolites points towards telling us that the microscopic anisotropy in the intracellular space is very, very high. Doesn't matter where you are in gray matter or in white matter. Doesn't really matter whether you are also in astrocytes or in glia or in neurons, both gray and white matter, with the exception of the choline in gray matter, which I put on the side with a big question mark on top. Oh, am I still okay, or shall I kind of wrap it up?
You have a couple of more minutes.
In that case, what would be more of interest, clinical results or functional results?
Whatever you prefer. I think some clinical data would be cool.
Okay, let's focus on clinical data, and the functional I will use in the next time I'll waste your lunch time. In the case of clinical stuff, let me go back to making this function. Okay, I'll skip the tortuosity. It's an interesting debate, but I think a little bit too spatialized at this point, also in the little time that we are left with. In case of clinical results, I think it's useful to see a cartoon that kind of tells us what is that we really expect from things to happen. It's not that we wait for this hypothesis to fulfill the prophecy, but it's a good idea to come up with, at least with the hypothesis regarding to what we expect to see happening. And one thing that one can imagine is that when we talk about pathological phenomena that affect glia and that affect neurons, they're quite different from one another. So for example, what we expect to see that affect a pathomechanism that would affect neurons is exemplified in this case in the neurofibrillar retangles in tau neurofibrillar retangles in AD. But one can think about any other intraneuronal or intraaxonal pathology in which structures within the intraneuronal space change their properties, and this change may affect the diffusion property of metabolites in the intracellular space. What direction? I'm not sure, but whatever will happen in the intracellular space, in the intraneuronal space, will affect the, let me go back to the laser pointer, will affect the metabolites that reside in those cells. In glia, the pathomechanism that would be expected to give the most, let's say, dramatic change in diffusivity of intraglial metabolites is what we call glial activation. In glial activation, I reserve also the possibility, by the way, of breakage of structures within astrocytes. So for example, in the disease called PSP, there is also accumulation of tau in astrocytes. But most of what we expect to see with respect to glia is glial activation in response to neuroinflammation. And that would cause changes in cytomorphology of glia that will, again, impact the diffusion, or we hope so at least, the diffusion coefficients of glial metabolites. In what direction? Well, that's a little bit of kind of telling the history backwards, but I hope that I convince you that for what we know, it does result in an increase in diffusion coefficients of glial metabolites. And I want to start with a couple of, well, I'll skip the historical background. There is a historical background here that shows experiments from the 1990s that actually show change of diffusion of metabolites in stroke from the labs of Jens Fram, but also from Klaas Nicola in Utrecht, and go to the first diffusion-weighted MRS, to the best of my knowledge, that was done on humans in clinical environment. And that's the experiment that was instigated from me by Danny Reich, now at the NIH, at that time in Johns Hopkins, together with his then PhD student, Emily Wood, in which we measured the diffusion properties of NAA and of water in the corpus callosum in a cohort of MS patients. in corpus callosum, normal appearing white matter. And you can see here a very, very interesting result. We were quite naive at that time. We measured the tensor of the NAA, well, assuming that we are relatively organized in the corpus callosum. We looked at the main eigenvalue of the diffusion tensor of the NAA in the corpus callosum and compared the cohort of healthy controls versus multiple sclerosis patients, again, normal appearing white matter. And to our surprise, the mean value for the axial diffusivity of NAA in the corpus callosum was lower in the MS patients than it was in the healthy controls. And that is in complete contrast to the same result measured with water. And this is, again, it's a huge debate that people have regarding longitudinal diffusivity. Does it measure solely axonal damage? Yes or no? Well, here is quite a strong evidence, if you want, that the axonal or axial diffusivity of water from the diffusion tensor imaging can be modulated more strongly by even slight demyelination that is happening even in normal appearing white matter, because this is quite hardcore evidence that, by the way, was repeated also in the longitudinal study that followed this study, that shows that the intracellular diffusivity in axons in MS actually is expected to go down. I will skip the longitudinal version to it. I can give you the reference if it is of interest to you. One of the most beautiful experiments to show that there is a link between neuroinflammation and diffusion of metabolites in the brain is results that come from the Cuprizone model, in this case, an animal experiment that was done by Francesca Branzoli and the group in Paris, helped also by Julien Valette and his group. And to those of you that don't know, the Cuprizone model is a model to induce reversible demyelination in a whole host of different types of animals, from rodents to marmosets. In this case, it is in rodents, in the mouse. And you can see that the measurement here is a diffusion-weighted measurement aimed at measuring what happens in demyelination in the corpus callosum of the mouse in the Cuprizone model or after administration of Cuprizone. And since it is a demyelinating effect, the effects that we are measuring in metabolites are expected to be seen in the intracellular space, regardless of the demyelination. So these are measuring something else. And what you can see is that indeed on the neuronal metabolites, there is simply no effect whatsoever. The diffusion-weighted values for NAA at any B value do not differ between the Cuprizone mouse and the control mouse. The glial metabolites, on the other hand, the choline compounds and marinositol, you can see a huge effect. This is the control mouse, and this is the Cuprizone mouse. You can see that in both cases, an extremely strong effect, and it is an effect that does not necessarily reflect the effect on concentration of these metabolites. So for example, there is a decrease in concentration of NAA, maybe due to neuronal death, but those neurons that survive do not change in their diffusion coefficients, which is a very curious and interesting finding. Or for example, there is no change in the concentration of marinositol between those two, between the mouse, the control mouse and the Cuprizone mouse, but the diffusivity of marinositol increased dramatically. So we have here an interesting evidence of an immune response that is causing the apparent diffusion coefficient of glial metabolite to increase, in this case, in a Cuprizone model. And in the next two slides, which I think will conclude my my talk to you guys, in one disease and one model for inflammation, induced model for inflammation in humans. Brace yourself. There is such thing, induced model of inflammation in humans, and I will talk about it in a second and will conclude the talk here. Evidence that we got for modulation of diffusion of metabolites in a neuroinflammatory disease came from a collaboration with the Department of Rheumatology here on a autoimmune disease called lupus. Many of you, I'm sure, heard about lupus. Lupus is a devastating disease that can have, not in all cases, but can have an effect on the brain. And when it has an effect of the brain, it can also result in neuropsychiatric complaints. Our hospital is a national referral hospital for lupus patients with neuropsychiatric complaints, and we recruited them after being, scanned for the regular clinical scans, also to be scanned on the 7 Tesla for diffusion-weighted MRS and diffusion-weighted imaging. So from the diffusion-weighted imaging, we ascertain the fact that we do have, or we do see, microstructural alterations in the corpus callosum of these patients. We stuck to the corpus callosum just out of habit, and we measured the diffusivity of those metabolites in the same region in which we saw also decrease in FA and increase in MD in the corpus callosum of these patients. And what we saw, to our surprise, is that NAA doesn't seem to be affected at all, but the diffusivity of creatine and choline seems to increase when you go from healthy controls to SLE or lupus patients without neuropsychiatric complaints to those that have neuropsychiatric complaints, which is an interesting finding that we think indicates, again, an immune response that is reflected in a selective increase in the diffusivity of glial metabolites, but not of neuronal metabolites. And I will conclude with this very, very interesting experiment that was instigated by Neil Harrison and Mara Cerciniani when they were both in Brighton, where I'm moving now. Neil Harrison, who is a neurologist or a psychiatrist with strong interest in neuroinflammation, looked at models of induced neuroinflammation that are used also in animals, but in some labs were also induced in humans. And one of the ways to induce systemic inflammation, not necessarily neuroinflammation, is to administer lipopolysaccharides intravenously to animals or humans. This is something that creates an immune response on a systemic level. And the hope was that it will reflect itself in some sort of way also in a response of the immune system in the brain. And lo and behold, that even in a very small cohort, and it's a very small cohort, it's 8 subjects that have been repeatedly measured as controls to themselves. So before administration of LPS and after administration of LPS, this graph shows you the difference in diffusion coefficient for NAA and creatine in 2 different, and choline in 2 different regions. Here in parietal white matter region, where we detected no difference in the diffusion coefficient of any of those metabolites before and after the LPS administration. But in the thalamus, there was a distinct and highly significant increase in the diffusion coefficient of choline alone, and not in creatine, which is also interesting to me. I don't have an explanation to it. Definitely something that has to be corroborated from different sources. And interestingly, this difference is not expressed in increase in or any change in the concentration of those metabolites. So again, going back to the Cuprizone model, we have 2 independent measures for what is happening in response or to insults to the brain, both on the neuronal as well as on the glial level. This measurement does not strip the concentration measurement from any meaning. It actually adds an additional vector in which we add additional information that collated with the measurement that we do on the concentration of those metabolites simply gives a more complete picture of how the pathological mechanism acts on tissue, both on the neuronal level as well as on the glial level. And I think that that would be a good time to do a very quick fast forward to the conclusions. I will skip the Lawrence workshop that I really wanted to tell you about, but I can talk to the people that are interested in that later on. So fusion-weighted MRS experiments, difficult, I can tell you. They are difficult, they take time, there is lots of post-processing that has to be done with a very careful manner and lots of quality control as well in the acquisition level, but also in looking at the data later on and making sure that you're not measuring garbage. And this is a kind of a general appeal. So if it is of interest to you, consider including this type of measurement in your protocols. One can do short experiments that focus on measuring ADCs, simply something like 10 or 15 minutes, or one can do for longer experiments, let's say, that can result in more detailed information like the diffusion, double diffusion encoding experiments that I mentioned earlier. And importantly, there is quite a community now of diffusion-weighted MRS people that are eager to help people both on preclinical scanners as well as on clinical scanners to do their experiments. Please come to us. We provide everything for happily and for free. So just get back to us if you are interested in this type of measurements. And thank you very much for your attention. And thanks very much all for inviting me to give this presentation.
Thank you so much, Dr. Ronen, for this great talk. It was a bit longer than expected, but it was worth it. Absolutely. So if anyone has any questions, so feel free to either use the Q&A chat or raise your hand, and we'll try and accommodate a single or a couple of questions. And while people are typing, I'll use just the option to satisfy my curiosity. How does it look when you apply this method on lower fields? I think mostly was on 70, but how does it look on 3T and even out of curiosity at 0.05T spectroscopy?
Well, 0.05T I will exclude from the discussion right now. I'm now, as Matt Rosen is struggling in getting spectroscopic data to begin with. This is a different topic that, of course, I'll be glad to talk about some other time because I really love Matt's results and I think that one can it's important to focus on that. At 3 Tesla or at 1.5 Tesla, again, it all comes down to your variability and what you're trying to measure. If you are interested, for example, at 3 Tesla, I can tell you we actually had a nice robustness and reproducibility study that was performed here at 7T and at the NIH on 3T, just focusing on a couple of diffusion-weighted MRS type of evaluations. And to be perfectly honest, when it comes to ADC, like for example, the LPS measurement is all 3 Tesla, there is no barrier to do those measurements on 3T. If your volume is big enough, these days, for example, a system like the Prisma scanner has such wonderful gradients that you can get to very high B values at a relatively short echo time. So whatever you lose from going from, whatever you gain, let's say, from going from 3 Tesla to 7 Tesla field-wise, you may lose with weaker gradients if you don't have a tera. If you have a tera, then okay, you're all set for life. But if you have systems like ours that have weaker gradients, then everything is balanced and it all ends. The question should be answered, what's your SNR? What's your variability in your end measure? And I can be quite optimistic about most of the things that we do at 7T can be done at 3T, easy, no problem.
Great, thanks a lot. And I think there's one quick question from Ahmad Razakan. Ahmad, if you could please unmute. And if not, I can read it. How much sensitivity and specificity of these metabolites, NNA and ML, with cells like neuron and glia? Yeah. And it's midnight on his time, so we can't speak.
Oh, okay. I see. How much specificity? Yeah, that's the million or billion-dollar question. Ovidio is here. I think he can give a much more learned response to this question than I have. I think that we're a little bit lazy in responding to this type of concerns. I think that, for example, the NAA question is not fully answered as well, because there are people that would say that there is an oligodendrocytic concentration of NAA that we completely ignore. I would respond to that. That might be true, because actually histology shows that there is NAA in oligodendrocytes. The question is, what is the relaxation time of NAA in oligodendrocytes if it's wrapped within the myelin sheath? And the question to that has to be answered in a systematic way. There is no pure answer to it. So the question has to be answered, I think, with the proof of the pudding is in the eating, in the sense that if you have an experiment in which you see a strong modulation or an experiment that you know that affects a specific cell population, and you see a result that is expressed in changes, for example, in diffusion properties of a set of metabolites, but not in the other, or less in the other, then take these data and build them in a model that includes concentration of metabolites across metabolites and their diffusion coefficient as well, in a kind of, let's say, combined model that takes all this information, use all the priors that you can have, not only on the distribution of metabolites across cell types, but also what you know from the disease or the pathomechanism that you exert, what effect it should have on tissue, and come up with an answer later on. I think that this is one of the ways to go. And the other one is really to be a little bit more proactive in constantly looking at models or newer models to try to truly characterize the concentration of different metabolites in tissue. Because I think that the truth is still out there, if to use the famous phrase from the X-Files, and we are a little bit too lazy in just saying, you know, glial metabolites, choline, myo-inositol, neuronal metabolites, NAA, and glutamate. So good question, calls for a more specific answer.
Thank you. And last questions from Ovidio. Can you please unmute?
Yeah, hi, Itamar. Thank you for this presentation. I learned a lot of things. So one thing that I haven't followed so much the literature in the field, but from your presentation is, and that's what I kind of learned, or it makes me a bit wonder. So you, from the data that you presented, the diffusion times, the diffusion constant that you measure depends on the time, the diffusion encoding time that you use, right?
Yes and no. Yes, I mean,
is that so? Using this, you can also probe different subcellular compartments. So let's say is this because also you have different I don't know, NAA pools, like pools of NAA that diffuse faster. Let's say some that are or slower. For example, if you'd have NAA which is packed in a more tighter compartment, is that also possible explanation or not? Or
we are hoping that this is an explanation. So it's an excellent question. So, for example, it is known that there is a sizable NAA concentration in mitochondria, and the question is the mitochondrial NAA enough to give this kind of time dependence that one would expect to see at some point, because mitochondria are very small structures. So I'm guessing that the time dependence will become flat or even non-existent at some point. But I don't know, for example, what are the relaxation times of NAA in the mitochondria? And the same goes for glutamate. For example, does the vesicular glutamate have any effect on either the spectroscopic measurement or the diffusion measurement? I think that as I answered to Ahmad earlier, this really calls for, let's say, a systematic investigation of this question, starting from in vitro systems, going through, let's say, systems, let's say animal systems, where you can control these things a little bit more or better than in humans, and then project into the human measurements. But all that you ask are very, very open questions. The question about glutamate, vesicular glutamate, it's something that I posed to a friend of mine in Germany, and you know, the Germans always have an experiment in mind to do in any conjunction. And he already had something that he already suggests. Yeah, we can do this, and we can get the vesicles in one side, and the so, okay, you come up with the experiment, do it, and I will publish the results. But I think it's an open question how subcompartmentation affects not only the diffusion measurement, but also the actual concentration measurements, because it's a multi-dimensional situation where the relaxation properties play in part of the game and the diffusion properties. And I really don't know what is the concentration within those subcompartments. All very interesting open questions.
Okay, thank you so much for all of the information.
Thank you.
So thank you so much again, Dr. Ronen. It was a great pleasure, and I think we don't have any other option than having you again sometime later this year for another talk on the rest of the topics we didn't get to hear.
If you're not too tired of me, then I'll be happy to join again. And I constantly visit Boston. As I told, I will be in Boston actually next week for 10 days. So maybe I'll just hop and say hello, and whoever is there, I'll be glad to meet and say hello.
Okay. I'm looking forward to meet you.
Super. Same here.
Okay.
Okay, guys, have a lovely afternoon. And thanks again for the invitation. Oh. And I'll hope to see you very soon in London as well, but hopefully in Boston earlier.
Absolutely. Thank you, everyone.